Good morning, everyone. I am your host and co-speaker for today, Liz. I'm an Associate Director of Product Marketing at PacBio. Today, I'm joined by Brian Haas, who is my co-speaker. He is a research scientist at the Broad Institute. We are going to talk about using his latest tool, CTAT-LR-fusion, for cancer fusion transcript discovery, along with using the PacBio sequencing and the Kinnex kits. A few housekeeping items before we get started. This webinar is being recorded. You will receive a recording in your email about a week after today's event. All attendees are muted. During any time in the webinar, you can use the question box, which should look like rectangular with a question mark on your upper right panel in the GoToWebinar. You can ask the questions at any time. However, we will only be answering them at the live Q&A at the end of the recorded talks. There is also the materials box. If you click on the upper right corner with the little paper sign, it will actually show some of the application notes for doing Kinnex single cell and Kinnex bulk RNA sequencing. So with that, we will begin the recorded talks, and we'll come back for the live Q&A at the end.
Good morning, everyone. My name is Liz, and I'm an Associate Director of Product Marketing at PacBio. Thank you for joining the webinar. Today, I'm going to talk about scalable, cost-effective RNA sequencing with Kinnex kits. PacBio HiFi sequencing are both long and accurate. It is as long as leading next-generation sequencing. Shown here on the left side, we show that with PacBio HiFi reads, you can reach over Q30 or 99.95% accurate. But it is also a lot longer. Typically, PacBio HiFi reads can reach 15 to 20 kilobases. HiFi sequencing has been applied to many different areas in genomics, from filling the remaining 8% gaps in the human genome to full-length RNA sequencing to understanding epigenomes. To understand why long-read sequencing is important for sequencing transcriptomes, here's the difference between using fragmented short reads and full-length reads from PacBio for differentiating isoforms. Can you tell how many isoforms there are? With long-read sequencing, we do not need to do transcript assembly, which is prone to errors. We get the entire full-length cDNA in one piece. For RNA sequencing, not only length is important for understanding the isoform complexity, accuracy is also important. In single cell, this might mean better detections of UMIs and barcodes, whereas for bulk RNA sequencing, having higher accuracy means better ability to call SNPs, indels, and detect junctions. Recently, the LR-GAS consortium published a study comparing different long-read sequencing methods. It showed at the time that PacBio had the longest reads and highest read quality, and that despite having 10 times less reads than ONT, it detected more genes with longer and rare transcripts. It also had a better ability to quantify isoforms. However, at the time of the study, PacBio did not yet have the Revio system or the Kinnex kits. Thus, it had the lowest throughput. This is no longer the case with the Revio system and the Kinnex kits. To look at how the advantages of long reads are also important for single-cell sequencing, compare the difference between short reads and long reads for looking at a cDNA transcript in single-cell libraries. With short reads, you are limited to 50 base pairs of the information. This gives you only gene counting information. No matter how much more you sequence, you will not get the isoform information. With long reads, the more you sequence, the more information you get. Recently, PacBio published the Kinnex kits, which are solutions for scalable RNA sequencing applications. There are 3 Kinnex kits. The Kinnex full-length RNA kit is for bulk RNA sequencing. On the Revio system, that generates 30 to 40 million full-length cDNA reads. The Kinnex single-cell RNA kits generates 80 to 100 million reads on Revio system. The Kinnex 16S RNA kit generates full-length 16S sequences that is suitable for species identification and generates 50 to 60 million reads on the Revio system. Various kits also support different kinds of multiplexing. We will now talk about the Kinnex full-length RNA kit. The Kinnex full-length RNA kit starts with cDNA synthesis using the Iso-Seq Express 2.0 kit, which generates up to 12 barcoded cDNAs that can be pooled together for multiplexing. Then, using the Kinnex array formation, different cDNA are concatenated into an ordered array into a longer fragment for hi-fi sequencing. The reads can then be bioinformatically deconcatenated to segmented reads or S-reads that represent the original cDNA molecule. The PacBio SmartLink software read segmentation and Iso-Seq workflow then processes the reads for primer extraction, isoform clustering, mapping, and finally, transcript classification with read count information. Having tested Kinnex full-length RNA kit across a variety of samples and tissues, we've seen the consistent trend that at around 10 million reads per sample, 80% of the known isoforms are detected. While the sequencing depth may vary by experimental goal, tissue, and species, it seems consistent that at 10 million reads for most samples, this would be adequate for genome annotation and quantification of medium-expressed genes. Here's an example of the WTC11 cell line detected at different depths. At 10 million reads or 4-plex on a Revio system, on average, for every sample, 17,000 genes and 47,000 isoforms can be detected. Using the USD list price, this means the all-in cost from cDNA synthesis to sequencing would be $488 per sample. We therefore recommend for most studies, 10 million reads or 4-plex on a Revio system is sufficient. For isoform discovery and comparison of highly expressed transcripts, we can go down further to 5 million reads or 8-plex on a Revio system. For plant and animal species, we recommend either 10 million at 4-plex or 5 million at 8-plex. We will now briefly touch upon the Kinnex single-cell RNA kit. Similar to the Kinnex full-length RNA kit, the cDNA used as input to the Kinnex single-cell RNA kit are single-cell cDNA generated either using the 10x Chromium single-cell 3', 5' kits, or the Parse Evocode WT kits. We target 3,000 to 10,000 cell recovery. There are barcoded Kinnex adapters to support multiplexing. And just like the Kinnex full-length RNA kit, SmartLink supports full end-to-end isoform processing and classification to generate SCONTI or Seurat-compatible gene and isoform count matrices. We've released public data sets to show that compared to matching short-read data, Kinnex single-cell libraries have high concordance in matching cell barcodes and UMI abundances, but that it also offers isoform information, generating cDNA up to 4 or 5 kilobases. To learn more about the Kinnex kits, visit pacb.com/Kinnex, or you can follow us on social media. Thank you.
Thanks to everyone for joining this webinar, and thanks to PacBio for allowing me this opportunity to present our latest work on fusion transcript detection using long reads. I'll begin this webinar with a short introduction to cancer transcriptomics and an introduction to our new method, CTAT-LR-fusion. From cancer transcriptome sequencing data, we can assess both functional and genetic readouts for tumors. We just need to resect the tumor sample, extract the RNA, convert it to cDNA, and then sequence it using our favorite modern sequencing technology. The transcriptome sequencing functional readouts include expression abundance, isoform structures, chimeric fusion transcripts, evidence of RNA editing, and insights into tumor cellular composition and heterogeneity. From the RNA-Seq data, we can further infer properties of the cancer genome, such as mutations, chromosome rearrangements, and copy number aberrations. To make the most of cancer transcriptome data, we need various computational methods and software tools. We've been developing software modules as part of our Trinity Cancer Transcriptome Analysis Toolkit to support various aspects of these research activities. We currently have tools and pipelines available for exploring cancer RNA-seq data, including transcript reconstruction, finding mutations, fusion transcripts, viruses, and copy number aberrations, and further supporting analyses for bulk or single-cell RNA-seq data. Much of our focus in recent years has been on fusion transcript detection. And this includes our development of our newest tool, CTAT-LR-fusion, for fusion detection using long isoform reads. Chromosome translocations and genome rearrangements can generate gene fusions, and certain gene fusions are leading causes of cancer. One of the best examples is one of the first oncogenic fusion genes to be discovered, which involves a chromosome translocation between the bottom arms of chromosome 9 and chromosome 22. This genome rearrangement yields chimeric chromosomes and fuses the BCR gene with the ABL1 gene, yielding an oncogenic BCR-ABL1 fusion gene. This oncogenic driver fusion is found in 95% of chronic myelogenous leukemia patients and is a hallmark of the disease. Whole genome sequencing can be used to detect structural variants such as this one, but it's expensive and doesn't tell you about the functional effects for the rearrangements. Transcriptome sequencing is much more routinely used to detect fusions because it's relatively inexpensive and directly captures evidence of the fusion gene products being produced. There are many gene fusions that have been found that are relevant to cancer. The COSMIC database currently lists over 300 fusions that are linked to cancer, and surveys of large cancer datasets have yielded thousands of potential candidates. Some fusions, like BCR-ABL1, are highly prevalent in certain cancer types and represent hallmarks of the disease. For example, SS18-SSX is a hallmark of synovial sarcoma, and TMPRSS2-ERG is found in 50% of prostate cancers. Certain fusions are clinically actionable, particularly those that involve protein kinases, as they can often be treated with kinase inhibitors and yield a more favorable patient outcome. Most of the earlier studies on fusion transcript detection involve traditional Illumina short-read RNA-Seq, and there are a couple of popular strategies for finding fusions based on these short reads. Reads can first be aligned to a reference genome or reference transcriptome, and those reads that align as split reads, with parts of individual reads aligning to different genes, or as discordant read pairs, where the paired ends align to different genes, are all identified as evidence for potential fusion transcripts. Another strategy is to first assemble the short reads into more complete transcript sequences, and then align the reconstructed transcripts to a reference genome or transcriptome, and identify those read alignments that are chimeric, with different parts of the read aligning to different genes and providing the evidence for fusions. In earlier work, we implemented methods based on both of these strategies. We built Star Fusion for finding fusions based on chimeric read alignments, and Trinity Fusion, which uses our Trinity RNA-Seq assembler tool to first reconstruct transcripts from the short reads before searching for chimeric transcripts. More recently, we published Fusion Inspector, a method for characterizing the sequence and expression characteristics of candidate fusion transcripts. Fusion Inspector models fusion contigs based on the fusion gene's order and orientation, and aligns the short reads to the fusion contigs to capture read alignment evidence and to define fusion transcript breakpoints. Fusion Inspector includes some downstream analytics and visualizations to help study the biological relevance of fusion isoforms, and these methods work with conventional Illumina RNA-Seq data. Now, we're in the era of long reads. Many advancements have been made in long-read sequencing over the last decade, with increasing throughput and decreasing error rates, both for PacBio and for ONT. And the latest stats and error rates are truly impressive. A key advance was made very recently for PacBio isoform sequencing with the development of MAS-seq by Aziz Al-Khafaji at the Broad Institute. This method was further commercialized as the Kinnex product at PacBio. This system is capable of yielding 40 to 120 million cDNA reads in a single run on the new Revio instrument. What's especially powerful about the system is that the long isoform reads can be generated for single-cell samples as well as for bulk transcriptomes. To make use of long isoform reads for fusion detection, we explored adapting components from our earlier work. Instead of using Trinity Fusion to assemble reads, we figured we could just use the long reads themselves. And we figured we could use components of Fusion Inspector for modeling fusion candidates, resolving fusion transcript breakpoints, and for the downstream visualizations. We call our new tool CTAT-LR-fusion, and it's the newest module of our cancer transcriptome analysis toolkit. In the initial Trinity Fusion-like stage, we identify candidate chimeric transcripts using a customized version of the Minimap2 alignment software, which we named CTAT Minimap2. It's 4 times faster than the regular Minimap2 because it only focuses on generating alignments for those reads that appear to be chimeric and map at multiple loci. Then in the second phase, for each of the fusion gene candidates, we model gene fusions by putting the genes of the fusion gene pair in the proper order and orientation, and realign the candidate fusion long reads to these fusion contigs. From these alignments, we define the fusion genes and fusion isoform breakpoints. The output of CTAT-LR-fusion includes an interactive visualization that allows you to navigate the evidence for the found fusions. This visualization tool is based on IGV reports and shows the reference gene annotations and the read alignments in the context of the modeled fusion contigs. Here, the full-length reference gene annotation structures are shown at the top, and the read alignments are shown below. From the read alignments, you can see which exons are spliced into different fusion transcript isoforms. If short reads are available in addition to the long reads, then Fusion Inspector is run, and the results are integrated along with the long reads into the report and the visualizations. The rest of this webinar is organized in 3 parts. First, I'll show the various ways we benchmarked CTAT-LR-fusion in comparison to alternative methods. Then I'll highlight the benefits of using both long and short reads together for fusion detection. And then I'll finish with some findings from our application of CTAT-LR-fusion to patient tumor samples at single-cell resolution. To benchmark and evaluate our fusion caller in comparison to other related methods, we explored a few different data sets, including both simulated data and genuine PacBio isoform sequencing data. For the simulated data sets, we used PVSim3 software to simulate both PacBio hi-fi reads and ONT reads. For genuine PacBio reads, we sequenced the commercially available fusion RNA standard made available by the company Seracare. We also sequenced the transcriptomes of 9 tumor cell lines in collaboration with the Cancer Dependency Map, or DEPMAP, group at the Broad. And finally, we examined the tumor single-cell transcriptome sequencing data generated for melanoma and ovarian cancer patient samples. The different long-read fusion detection methods that we compared include our tool, CTAT-LR-fusion, Jaffa-L, LongGF, Fusion Seeker, and PB Fusion. For benchmarking, we scored true positive and false positive fusion predictions based on truth sets and evaluated this as a function of minimum fusion evidence read support. Based on the numbers of true positives and false positives, we computed precision and recall metrics, for which then we generated F1 scores as overall accuracy metrics. We further evaluated the F1 score as a function of minimum read support and compared the peak F1 score for a given predictor. We performed benchmarking at varying levels of stringency. Given a fusion gene pair, as shown here where the purple gene A is fused to cyan gene B, we consider the gene pair ordering as reported by different methods. For benchmarking, we evaluated strict gene ordering, requiring the fused genes to be reported in the order in which they appear in the true fusion transcript, or we were less stringent and allowed the fusion gene pair to be reported in either order. We also evaluated fusion transcript breakpoint resolution accuracy, where we required the exact breakpoint to be reported correctly, or we were less stringent and allowed for fuzzy breakpoint detection, scoring a true positive if the breakpoints fell within a short distance from the known breakpoints. Here are the results for benchmarking fusion detection accuracy using simulated PacBio reads and exploring the different evaluation criteria across predictions, including requiring strict gene ordering, allowing for the reverse gene ordering, requiring exact breakpoints, or allowing for fuzzy breakpoints. Interestingly, only CTAT-LR-fusion and Jaffa-L yielded fusion transcripts with both accurate gene ordering and precisely defined fusion transcript breakpoints. If we ignored gene ordering and allowed for fuzzy breakpoint detection, then all the methods performed reasonably well on these simulated data. When testing with simulated ONT fusion reads, we essentially got the same results. Getting the fusion transcript gene ordering correct is actually very important and has serious consequences for clinical reporting and relevance to cancer. As a motivating example, here we have two genes as they're found in the human genome, TAC3 and FGFR3. These genes neighbor each other on chromosome 4. If we find a fusion transcript involving this gene pair where TAC3 is at the 5' end of the fusion transcript, we might consider it just resulting from read-through transcription, involving cis splicing, and not requiring any somatic structural rearrangement, and maybe not worth reporting. However, if we find the fusion with FGFR3 at the 5' end, this would require some structural rearrangement that would reposition the FGFR3 gene upstream of TAC3. This fusion is a known oncogenic fusion and would be essential to report, particularly in clinical applications. So when reporting fusions, it's essential to consider the order and orientation of the genes involved in the fusion. In addition to the simulated data, we benchmarked fusion transcript detection using real data. The company Seracare sells a fusion RNA mix that spikes in known concentrations of 16 different oncogenic fusion transcript RNAs into a background of a normal cell line RNA, and it's aimed for use as a standard in fusion screening efforts. We sequenced the Seracare fusion standard using MAS Iso-Seq and also Illumina TruSeq, both in triplicate. We had more PacBio than Illumina data, and so we downsampled the PacBio reads to match the number of Illumina sequenced bases. With CTAT-LR-fusion, we robustly detect each of the 16 control fusions, most with around 10 to 30 long reads. With the Illumina short reads, we find evidence for the fusions across all samples, but not all replicates across all samples. And when we do find evidence, it's usually just 1 or 2 reads. And so clearly, with the long reads, we're much more effective at identifying these control fusions than with the short reads. We examined the long reads using the other fusion predictors and found that several of the other tools, with the exception of the current PB Fusion software, failed to detect all the control fusions, and this was consistent across the replicates. To further evaluate fusion detection with long reads, we performed MAS Iso-Seq to sequence the transcriptomes of 9 tumor cell lines in collaboration with the DepMap project. These cell lines were selected because they harbored known fusions across diverse tumor types and were representative of a variety of different cancer types, including lung, breast, prostate, and blood cancers. Shown here are the counts of fusions predicted across cell lines by the various methods. Some cell lines, like VCAP, had many fusions predicted. Others, like MJ, had very few. Most of the methods had similar numbers of predictions for each cell line, except for Fusion Seeker, which tended to overpredict. There was generally good agreement among fusions predicted by the different methods. Numbers of fusions predicted in common by at least 2 methods is shown by the thin black lines. The known oncogenic fusions were identified by all the methods, thankfully, and the number of long isoform reads found as evidence were largely consistent across methods, ranging from only 2 reads to near 500 long fusion reads as evidence. Benchmarking fusion prediction accuracy using real data is more challenging than with simulated data because the complete set of true fusions is not perfectly defined. Here we used an approach where we defined a proxy truth set containing all fusions that were predicted by multiple methods, or uniquely predicted but separately found to have support from Illumina reads. Altogether, this gave us 145 proxy truth fusions. In benchmarking fusion prediction accuracy with this proxy truth set, we find that CTAT-LR-fusion maintains the highest accuracy across a broad range of minimum long read evidence thresholds. Next, we turn to harnessing the power of both long and short read RNA-Seq for fusion detection. For some fusions, we find evidence for alternatively spliced fusion transcripts, and this is supported by both long reads and short reads. As an example, this CYTH1 EIF3H fusion is found with 5 different splicing isoforms, all involving the first exon of the CYTH1 gene spliced into 5 different exons of the downstream partner EIF3H, with alternative splicing isoform breakpoints as shown by the green arrows at the top. In the case of this fusion, there's a nearly perfect correlation between the amount of short reads and long reads supporting each fusion splicing isoform, as shown on the left. On the right, we have the fusion expression information plotted in the context of the reference gene structures, with the size of the point reflecting the amount of read support. And curiously enough, for this example, we see a decrease in fusion isoform usage according to the position in the EIF3H gene. We find other examples of fusion alternative splicing supported by both long and short reads, and they aren't nearly as correlated as with CYTH1 EIF3H. Most show a consistent trend, but in some cases, the apparent dominant isoforms can be quite different. By leveraging both short and long RNA-Seq reads with CTAT-LR-fusion, we can maximize our sensitivity for detection of alternatively spliced fusion isoforms. For example, in this TMPRSS2 ERG fusion, where we find 4 fusion isoforms based on long reads, only 3 of them were detected using short reads, with 1 missing as shown by the X. In the case of TAT-DN1 GSDMB, there are 13 fusion splicing isoforms, all of which were detected from short reads, but 4 isoforms were missing from the long reads in this case. Ultimately, by using both short and long reads together, we're able to get a more comprehensive view of the fusion isoform diversity in these samples. In comparing fusion isoform expression levels across all fusion isoforms based on either short or long read support, we find a significant positive correlation, but there are very notable outliers. By ranking the fusions according to short read enrichment, you can see that most of the points fall below the center lines, indicating that long reads generally provide better support for fusions than short reads. But again, there are clear exceptions. One very notable exception is the BCR-ABL1 fusion that is the hallmark of chronic myelogenous leukemia, where it's detected about 100 times better with short reads than it is with long reads. We figured the difference in detection of fusions by short versus long reads is likely related to differences in sequencing strategies by the two approaches with bulk RNA-Seq. With short read bulk RNA-Seq using the Illumina TruSeq approach, the RNA molecules are first fragmented into smaller pieces that are on average a few hundred bases in length. Each of these fragments then becomes a separate target for cDNA synthesis, and so you get short cDNAs, but coverage across the entire length of the transcript. With MAS Iso-Seq, cDNA synthesis begins at the very 3' end of the transcript, and ideally you get the entire RNA molecule. But for very long RNAs, it can be difficult to get the entire molecule reverse transcribed, presumably due to the processivity of the reverse transcriptase enzyme. Hence, it can be challenging to obtain and sequence very long cDNAs. One thought was that the further the fusion transcript breakpoint was from the very 3' end of the RNA molecule, the more difficult it might be to detect it from long read sequencing in comparison to the short read sequencing. We explored this further by taking all fusion supporting reads and examining short to long read support as a function of the breakpoint distance from the very end of the read. We did find a weak positive correlation that was significant, but BCR-ABL1 continues to be a huge outlier. And so while the distance from the 3' end is likely partly responsible, it doesn't fully account for what we're seeing with BCR-ABL1. And so we'll be continuing to explore this later on in collaboration with DepMap, especially as we start exploring hundreds of additional cell lines with MAS Iso-Seq. In this last part, I want to highlight our fusion detection capabilities using long-read isoform sequencing with tumor single-cell transcriptomes, which is something that's particularly challenging to do using short reads alone. For single-cell transcriptome sequencing, a popular approach is using the 10x Genomics Chromium platform. This approach encapsulates a special type of bead with individual cells and droplets, and each bead has a cell barcode and a poly-A tail that captures the RNA molecules from the cell. Reverse transcription generates cDNAs, and then the sequencing can be run via Illumina or PacBio, getting short or long reads. But here, both the short and the long reads originate from just the very end of the transcript molecule. To demonstrate the value of CTAT-LR-fusion and long-read isoform sequencing on tumor samples and for single-cell studies, we turned to earlier published data sets from our own studies and from those of our collaborators. First, we explored a T-cell enriched melanoma patient sample from a study published last year, where we had 20 million PacBio mass Iso-Seq reads and about 200 million Illumina 3' reads from a 10x experiment. This involved about 7,000 cells, where 10% were cancer cells and the rest were infiltrating T cells of various types. We applied CTAT-LR-fusion to the PacBio long reads and fusion inspector and star fusion methods to the Illumina short reads. We identified only a single cancer-specific fusion involving NUT M2A AS1, which we found to be a known oncogene. A few of the normal T cells were found with short read fusion evidence, but it could have easily derived from ambient RNA. Only cancer cells were detected with this fusion using long reads. The vast majority of the cells identified with this fusion had only long-read transcript evidence, underscoring the value of long-read isoform sequencing for fusion transcript detection with single-cell data. Also, looking at the transcript structures that were resolved from the long reads, we find 8 different alternately spliced fusion transcripts, and most reads appear to be full-length. In contrast, from the short Illumina reads, we found evidence for fusion alternative splicing, but the fusion reads only identified transcript breakpoints rather than providing the longer isoform structures. In another tumor sample we explored, corresponding to high-grade serous ovarian cancer, we had 54 million PacBio isoform reads and 35 million 10x 3' reads, corresponding to about 500 single cells, with 20% of them cancer cells, and we detected 4 cancer-specific fusions. Shown in the UMAP plot are the cells colored according to cell type, and at the top we have the cells defined as cancer cells, with 2 of the most prevalent fusions indicated by the black dots. These 2 different fusions showed up on opposite sides of the cluster of tumor cells, suggesting that there was some tumor heterogeneity. Focusing on just the tumor cells, we find that there is evidence for 2 largely distinct clusters of cells. All 4 of the fusions we discovered are highlighted here, and there does appear to be differences in fusion isoform expression between these 2 clusters. Interestingly, 3 out of the 4 fusions we found were only discovered using long reads. In summary, we developed a new fusion detection tool called CTAT-LR-fusion. First, we stressed that fusion gene order, orientation, and breakpoint resolution are essential for clinical applications, and this was a primary focus of CTAT-LR-fusion development. We showed that CTAT-LR-fusion excels at long-read fusion detection, which we demonstrated with simulated reads, the Sarah-Cara fusion standard mix, and depth-map cell lines that we sequenced using MAS-Seq, or Kinnex. We further demonstrated that by using long and short reads together in CTAT-LR-fusion, we can further improve our detection of alt-splice fusion isoforms from single samples. And finally, we highlighted several of the merits of long reads when used for single-cell transcriptomes, including detecting more cells with fusion reads and resulting full-length isoforms as opposed to partial splicing patterns evident from short reads. Our CTAT-LR-fusion software is freely available as open source on GitHub, where we also have documentation and sample data. All the experiments shown here in this webinar are more thoroughly described in our preprints on bioRxiv, which is currently under review. And finally, I'd like to acknowledge those involved in this work, including our methods development lab at the Broad Institute, led by Aziz Al-Kafaji, our DepMap collaborators, led by Francisca Vasquez, and I'd like to point out Alvin Chin, who was my main partner in the development of CTAT-LR-fusion, Victoria Popik, who's an independent PI at the Broad Institute, and our collaborators Nico Barenwinkel and Arthur Dondi from ETH Zurich. Thanks again to all the attendees, and I'm happy to answer any questions.
Thanks, everyone. So if you have any questions, please use the questions chat box. We already have some questions coming in. I'll answer 2 of them. Yes, this webinar recording will be available afterwards. It will be available through your email inbox, and we usually put them on PacBio's YouTube channel as well. And there is another question that I think it's for me, is have you had the opportunity to compare Kinnex with the Tequila-Seq method published by Chop UPenn last year in terms of efficiency and reagent cost? So my understanding is Tequila-Seq is a targeted enrichment using like twist probes. I think this is a generic question not related to cancer fusion specifically. If it's simply referring to is Tequila-Seq comparable to Kinnex, I want to say that the Kinnex full-length RNA single-cell kits are by nature a whole transcriptome sequencing application kit. However, you can do targeted Kinnex bulk or single cell on top of that. In fact, I will say that we are working with different partners to generate official protocols that will make targeted enrichment in both bulk and single cell compatible with the Kinnex workflows. But no, we have not done a direct comparison. I think to do so would require saying, are we going to use the same Tequila-Seq probe? So I'm curious for the person asking this question, is like, what are you thinking in terms of, are you doing targeted to target enrichment comparison or just targeted to whole? So yeah, I don't have any direct comparison at this point. Okay. Okay, so let me ask some of the questions. This one is for Brian, I believe. Is CTAT-LR-fusion applicable to data generated before Kinnex?
The data generated before Kinnex?
So yeah, I think it's like, regular Iso-Seq, single cell Iso-Seq. It's not like
concatenated. Any long read isoform sequencing data should work. Could be ONT data, PacBio data, regular Iso-Seq. Kinnex. I mean, the big benefit of Kinnex is that you're getting many more reads for your sequencing output, which is hugely useful. But yeah, any long read isoform sequencing data should hopefully work.
Sounds good. Here's another question for Brian. Would the use of CTAT-LR-fusion to detect fusions not in cancer but in other conditions and even healthy cells that have much lower fusion expression be available? And actually, I think an even better extended question is, what is the minimum sequencing depth you would recommend?
Okay. So you can detect fusions in normal tissues just as well as you can in cancer tissues. We annotate them as being found in normal tissues, at least based on our earlier surveys of GTEx data based on short reads. But yeah, any fusion transcripts that it finds, it could be due to rearrangements, could be due to just trans splicing. There's natural structural variations in the normal human population where there are some samples that will show fusion transcripts that are not in other samples, and it's entirely normal. These will show up routinely in samples. And what was the other part of that?
Sequencing depth. And I think that's
a
very valid question, not just for fusion, but not for cancer samples, but any samples, like what's the sequencing depth required?
Right. So we, for most of the samples that we studied, we were able to do just, a few million reads per sample on Kinnex. So usually it's like between 3 million to 5 million reads per sample. As far as like doing filtering, based on our earlier work with Illumina, we required a minimum of 1 read per 10 million total reads. So 0.1, what we call fusion fragments per million total reads.
Okay.
But in long reads, especially, you can use a filter that's 10 times higher than that. So usually use like 1 FFPM, 1 fusion fragment per million reads as our like lower end. That basically matches up with 0.1 FFPM. So yeah, just a few million reads is fine. I mean, the more reads you do, of course, the more sensitive you're going to be for being able to find fusion transcripts. And if you're doing single cell data, then typically we'll do many more reads. We can do even up to like 100 million total reads. But because of the single cell Kinnex, you're doing 16 times as many reads as you would normally do because of the 16x array size. Every read is basically 16 cDNAs that are stitched together. So yeah, so you can get like 100 million reads for a single cell sample, and that works pretty well.
Cool.
Because that way you're not looking for just one fusion, right? You're looking to find that fusion in many different tumor cells or other cells, and be able to compare them and doing your single cell analysis. So having more reads is definitely going to be more helpful.
Cool. I think this is a question that's on the short read fusion side, which it would be Fusion Inspector, is when incorporating, or actually I guess it's like when incorporating the short read data, how is it determined that a short read belongs to the fusion or the wild type? Are only chimeric reads used?
Yeah, so for Fusion Inspector, if you have a read that aligns partly to both genes or exons of both genes, that would provide you with evidence for the fusion. If the read is just aligning to the one gene and not the other, then you'll see that it's actually quantified separately. So Fusion Inspector, based on the short reads, it'll tell you what your fusion allelic ratio is by looking at the fusion breakpoints and examining the reads that provide evidence for the fusion in comparison to the reads that do not provide evidence for the fusion. They actually overlap the breakpoint and align concordantly to that gene.
I think that actually is answering the other question I was looking at, because this question asked with short read, how do you distinguish reads that support the fusion when they don't span? And I think you're looking at that ratio. Can I ask, maybe extended, you were looking at some of the quantification. So, what's the formula for quantify? For long reads, it's a little easier. It's like, it's interchromosomal or, far apart. For short reads, how are you quantifying it?
I mean, it's basically using the same evidence for quantifying short reads or long reads. That you're looking for reads that provide specific evidence for that fusion, that breakpoint. With short reads, you can have two kinds of reads. You got the split reads, where individual read actually provides evidence for the breakpoint, or you have the spanning reads that are discordant reads, where you don't have the actual breakpoint information, but you just know that the reads are aligning to different genes. With your long reads, we're not doing paired end reads with long reads. Long reads, you get the whole transcript, and you're basically every long read that provides evidence for fusion is going to be a split read. It's going to provide you with evidence for that breakpoint. And for quantification, we're just counting up the number of reads that provide us with evidence for the fusion, and we do a normalization for the total number of reads in the sample. Right, because as you sequence more deeply with the fusion expressed at any given expression level, you're going to get more reads for that as evidence for that fusion. But again, there's this disparity between, so when we quantify, we use this FFPM statistic. It's fusion fragments per million total reads, and with short reads, you're going to have just a fraction of the reads that align to a gene or to that fusion, providing evidence for that fusion.
Yeah.
Right? Because the reads are short. Now, for the long reads, that whole read is going to be, the fusion read. So we'll generally find, 10 times fewer fusion reads per million sequenced with the short reads than we do with the long reads.
Okay. I think that partially explains some of the figures that I saw where in the SeraCare example, I was curious why the short reads, the numbers were so low, and I think it's because you were limited by the number of reads that actually cross the.
Well, there it's a little different because we tried to, we downsampled the PacBio reads to reach the same sequencing number of bases.
Okay.
Right. So if you're looking at like number of fusion evidence reads you have per number of sequenced bases, that should be pretty similar between the short reads and the long reads. So we kind of expected that we're going to see similar numbers of reads in that SeraCare example, because we had the experiment configured so that we had the same number of sequenced bases instead of the same number of reads.
I see.
Yeah. So there's something interesting about the SeraCare fusion standards. It turns out that those, by looking at the actual reads that were sequenced with PacBio, they appear to be just a little over 1 kb, like 1 to 1.1 kb in length. So they're relatively short. They're not like full-length fusion transcripts that we go end to end. They're designed to be a fusion standard, and they're larger than just like a couple exons stitched together. But they are only like around 1 to 1.1 kb based on what we sequenced. And so it might be just that's sort of they're like preferentially integrated into these MAS-seq arrays for sequencing. I mean, we don't really know, but there's something about the short read versus long read that is preferentially sequencing these fusions with the long reads. So it was kind of surprising. Yeah, we don't have a great explanation for that.
Got it. Here's another technical question. When you call a fusion, what do you call a fusion on something that merges gene A, which is annotated to, let's say, an intergenic sequence? So do you only report ones in which there are gene annotations?
Yes, yeah, that's right. So that's actually it's one of the, I don't know if it's like a pitfall, but it's a caveat, I guess I should say, and that you're only going to get fusions reported where there is going to be overlap with reference exon annotations. And actually, in our study, there was an actual novel transcript in one of the single cell samples that we were not able to detect because one of the fusion pairs was not actually in the reference genome annotation. So there's ways to compensate for that. Maybe in some future version of the software we might do that, but at least for now, it really does require that you have overlap with the reference gene annotations.
Got it. Here's a great question, is that, of course, you and I are very familiar with the MAS-seq Kinnex concatenation method. So a legitimate concern is, would you be accidentally calling artifactual fusions when the concatenation wasn't successfully deconcatenated?
Oh yes, that's actually that's a good question. You probably could. I haven't seen examples of that with CTAT-LR-fusion.
Yeah.
Probably because with the fusions that we're calling, they're not like they're two complete transcripts that get stitched together. Usually it's part of one gene and part of another gene, and the breakpoint that you find involves splicing of the two exons together. So it's a very different kind of fusion that you would find and report. Or if you have two genes that are like concatenated together into a fusion, kind of like an operon kind of situation, right? Or polycistrone, or whatever you want to call it.
Polycistronic.
Yeah, polycistronic transcript. Then that would look very different. You wouldn't have the breakpoints at splice sites between the two genes.
You're right.
Like
It wouldn't have a break.
and
Yeah, it
the
wouldn't have a break point. I
yeah.
see what you mean. Yeah. Okay. So maybe there
Some
are,
tools
but
could still report it though. Our tool probably would not report it. Yeah, I have to look at some good examples, but I didn't actually find examples of that when in surveying the results of our different experiments.
yeah. And I do want to say, add on to this question, is since this is also touches on the product development, is during the original MAS-seq and the subsequent Kinnex product development, we were very careful about bioinformatically being very aggressive with deconcatenating, and that is exactly because we do not want to accidentally call fusions. I think I wouldn't be surprised. There's always some really low amount that isn't successfully deconcatenated, but actually, if you look at the Kinnex or MAS-seq reports, most of them are full arrays, and the fact that you can call 16 of them, these are the nice property of MAS-seq and Kinnex is they're ordered. The concatenators are very well known, so it's very unlikely that we would see a perfectly ordered array and yet somehow we didn't deconcatenate something, resulting in resulting in an artifactual concatenamer.
Actually, there's something related to that. So and that might be one of the differences with real ONT data that we've been playing with more recently. You do see more of these kinds of other fusion artifacts, I would say. If you limit your CTAT-LR fusion results to those that involve reference splice junctions, then it'll discard a lot of these other kinds of artifacts. So in the output, it'll tell you whether or not the breakpoint is consistent with the reference annotation exon structures or not. So it's a really easy way to filter those. But again, with PacBio, like you said, Liz, the Kinnex, they're very careful about separating out, like all 16 in an array would be partitioned out into 16 separate cDNAs. And that's probably why we didn't see that.
So here's a question that maybe is for both of us: are there recommended tools for calling fusions from long-read DNA-seq data? My feeling is you're really looking at chromosomal rearrangements, right? So you're not really looking at our transcripts, and there's been papers doing breakpoint. I think that's more like structural variations. But I don't know if Brian, you have other thoughts on this.
Yeah, I'm sure that there are tools that exist for doing that, but I haven't been exploring them because we're really focused on the transcriptome. It's kind of interesting. I mean, some of the tools, other tools that work for transcriptome long reads, they kind of treat the problem more like a structural variation problem, I think, than they do fusion transcript detection kind of a problem. But the other thing is that your breakpoints that you get from doing long reads, they're not going to be, they're going to be within intergenic regions, they're going to be within introns. They're not going to a lot of times fall within exons of reference genes, and that's what most of these fusion transcripts that we're finding involve, fusion breakpoints that are between two expressed transcripts.
Here's another great question. Can CTAT-LR-fusion handle complex fusions? When I first saw this, I thought it was making like 2-hop or 3-hop, but maybe I'll ask you that. But there's another one, like things like have inversions or translocations. So things that aren't just like you showed examples, it's very clearly 2 genes. But what about something more complicated than that?
It can do, I mean, any kind of rearrangement that, genome rearrangement that would result in a fusion transcript should be reported by CTAT-LR-fusion. In terms of like really, it won't report fusion transcripts that involve many, breakpoints as like a series of breakpoints and one result. You might get separate, gene pairs that result from different fusion breakpoints. But it's not going to isolate those reads that might provide for very complex, structural or like when the genome gets, in cancer, you find these regions like chromotripsis or thripsis, where it's like shattering of the genome and it gets put together in like random ways. And you might find some fusion transcripts that involve several different genes. That's not something that we focus on. We could, I mean, there are other tools out there that believe, highlight that as an example of where they can perform. But it is more complicated. I think most fusions that we know of in cancer, that are associated with specific cancers, they don't involve these really complex structures. But there are, token examples of these things that show up from time to time where we have very complex fusion transcripts. But we're mostly focused on just gene pairs.
I guess this is an extended question like that is, can it detect things like recombination of tandem repeats, so like very repetitive motifs occur in 2 genes, assuming it's a 2-gene fusion?
None of the tools, so we looked at things like how well does it work with repetitive sequences. So I actually focused on looking at paralogs and just simulating a bunch of fusions between paralogous sequences, and it doesn't do great. Sensitivity was not very high. It was maybe like at 50% or 60% or something like that. But none of the tools really did all that great. It's a challenging problem. So yeah, if you have fusion transcripts that involve genes that are very similar to each other, then it's going to be a more challenging problem, and I'm not sure that there is a really fantastic solution. Also, I'm not aware of good examples, at least in oncology, where the fusion that is like the driver fusion involves a pair of genes that are close paralogs, like nearly identical in sequence. There might be one that's been reported before, but the ones that I'm familiar with, I don't know of any.
Okay. Here's a question: have you had a chance to compare the performance of different long-read technologies for fusion detection?
Yeah, just PacBio and ONT are the only 2 for long reads. And again, with the ONT sequences that we've been studying, there was more fusions that we're reporting that don't involve proper splicing between 2 genes, and a lot of that could involve artifacts of various types. So I wouldn't say that we're optimized for ONT. Like, we know it works with ONT, and we can provide guidance for how to use it with ONT. A future version of our software might be better tuned for it, but honestly, we're focusing on getting everything to work well with the PacBio data.
We have a very specific question from probably one of your slides. Were there any patterns in the fusion expression in the single cell data for the NUTM2A-AS1 RPWV1 203L2.4 fusion? I'm sure you show that example somewhere in your slide deck.
Oh yeah, no, there's the melanoma tumor sample. It's basically like the one fusion that we found, and it was found in a lot of cancer cells. Was there anything more interesting about it? I don't think so. I mean, it was relatively highly expressed, and we, it was an interesting find when, because I had no idea what this fusion was, and that I basically just, you go to gene cards or just Google the gene symbol to find out, is there anything known about it, related to cancer, do a PubMed search, and just there's a couple papers that were published just within the last couple of years that showed that NUT M2A AS1 was relevant to cancer, right? It's a link RNA, and it's tied to certain cancers. So I thought that was really interesting. We found this one melanoma sample. But beyond that, I really don't know much more about it. Is it really involved in cancer for that patient sample? I don't know. But it was a really useful example for highlighting the different aspects of long-read fusion finding as applied to the single-cell data, particularly in that we can get these beautiful, full-length fusion transcript isoforms, which I think there was like 7 or 8 of them that showed up. Different splicing patterns, but all for that one fusion transcript. Yeah.
You had showed that you were able to compare it to, I think it was the DepMap, and here are some known fusions. Where is a resource or repository of like all the known gene fusion reference files?
We have, so that's a tricky thing. I mean, there's a couple different resources out there. There's the Chimer Pub, Chimer-Seq, Chimer Knowledge Base. They're all kind of tied together. And there's a few papers that describe that. So those are good resources. We use those. We use data sets that we compiled during our earlier work, because we have data sets like we ran for short reads. We ran all the TCGA samples through, all the GTEx samples through as part of the Fusion Inspector paper. So in the Fusion Inspector paper, we targeted over 30,000 different samples across a bunch of different databases. So that's one big resource. The supplementary table in that paper is actually a pretty good resource. But we have this tool called Fusion Annotator, and if you just Google Fusion Annotator, I'm sure it'll bring you to our GitHub website. And with that, there's a database of all fusion transcripts that we've cataloged from all these different databases like Chimer Knowledge Base and Chimer Pub, our own data sets, some papers that were published that did their own surveys. We incorporated those as well. So if you have any fusions, and so there's a whole data set there that you could simply use that database that we compiled, or you could just go to these different sources that we list on our wiki for that Fusion Annotator human database. We list all of our different resources for that.
Thank you so much for all the questions, and thank you, Brian, for answering them. We are at time, so thank you for joining the webinar, everyone. When you leave the webinar, there will be a survey pop up. We would really appreciate if you can fill in this survey. And with that, thank you and have a good day.